Hello, everyone. I'm Pankaj Garg, a senior scientist in the antibody engineering team at Twist Biopharma. And today I'll be doing a virtual poster presentation on our high throughput platform to develop highly potent and functional antibodies against G protein coupled receptors. So let's dive right in. GPCRs are 7 transmembrane proteins that are targets of almost half of our current drugs, mostly small molecules though. They provide an opportunity in a variety of disease areas, including oncology, infectious diseases, migraines, and many others. Now, thinking of antibodies as drugs, they provide additional opportunity compared to small molecules. For example, they provide selectivity, effector functions, and much more. But currently, there are only 2 GPCRs against which antibodies have been approved in the market, which shows that GPCRs are vastly underexploited by antibodies. One of the reasons being that they have restricted extracellular matrix. So what we do know about GPCRs is that they have diverse ligands that bind to this extracellular surface. So we at Twist Biopharma decided to use this binding information to design an antibody library that was focused towards GPCRs. Now, let's look at how we use this binding information to design an antibody library that was focused towards this GPCR family. So to do this, we leveraged Twist's unique capability to print over 1 million oligos on a single chip. As shown on the left, in a traditional approach, when we use a 96-well plate, we can only print 96 user-defined oligos. In contrast, as shown on the right here, in our silicon-based chip approach, we have 9,600 clusters on a single chip. Not only that, in each of these clusters, we can print 121 different oligos. So if you do the math, 121 times 9,600, which gives us over 1 million unique oligos that we can print in a single run. Each of these oligos can be about 300 base pair in length. So we can use these oligos as building blocks for an antibody library. And as antibody engineers, we can really benefit from this technology because it gives us the unique capability to incorporate a vast user-defined diversity in the CDRs. So now let's dig deep into our GPCR-focused library design. So to define the CDR diversity that is focused towards GPCRs, we mine all known binding partners to the extracellular region of this receptor family. This includes chemokines, helical peptides, loop peptides, GPCR extracellular domains, extracellular loops, anti-GPCR antibodies. And we assembled all of this data and designed oligos that were printed and incorporated into the heavy chain CDR3. Limited diversity was also incorporated into the other 5 CDRs. Now, since our library is designed to have long heavy chain CDR3s, much longer than what is observed in heavy chain CDR3s in a human immune repertoire, we've picked framework regions and stems that actually allow for these long heavy chain CDR3s to be displayed. We've QC'd our library by next generation sequencing, and the data from which was used to generate the heavy chain CDR3 length diversity plot that you see here. Our final library has about 10 billion unique scFVs displayed on phage. We believe that this design will not only give us the unique capability to identify antibodies against GPCRs, which has been very challenging so far, but it would also help us identify antibodies that bind to functionally relevant motifs, which is, I think, more critical for any drug target. So we've carried out POC studies using this library against a wide variety of GPCRs. And let me walk you through a discovery workflow and our POC data that we've collected against GLP-1R, a receptor involved in glucose homeostasis. So we usually start off by building a GPCR overexpressing cell line. In this example, I'm showing you data from our GLP-1R overexpressing cell line. So once we have a cell line that shows good cell surface expression of our target, we use it in cell-based selections. Our cell-based selection scheme is shown here on the bottom chart here. So what we do is typically is like we take our target overexpressing cell line and incubate it with our GPCR-focused library. The non-binders are washed off. The binders are eluted and amplified for subsequent round of selections. We typically also amplify the output VH pools for each round and sequence them on our MiSeq platform to monitor clone and enrichment and heavy chain CDR3 diversity over the rounds of selection. So this is an example data we get from our successful screening campaign. After selection, we pick about 1,000 clones from round 4 and round 5, which are sequenced, and the unique clones are reformatted to IgGs. So these thousands or so reformatted IgGs, or we usually typically get about 100 unique IgGs, and we these reformatted IgGs are purified and tested for binding in our FACS experiment. The binders from our FACS initial binding study are then characterized in functional assays. Here, I'm showing an example of a GLP-1R antibody that was developed using this library, and which shows that it specifically binds to our GLP-1R overexpressing cell line and acts as an agonist in a cyclic AMP assay. So for our functional hits, we usually also map our heavy chain CDR3s to identify known motifs. And in this example against GLP-1R, we observed that many of our functional antibodies had a GLP-1 motif, a natural ligand of GLP-1R. In addition to the data that I've already shown you, we also do a lot of further downstream characterization of our antibodies. One such example is a BVP assay, and our antibodies, although have long CDRs, seem to behave very well in a BVP assay. In conclusion, I've shown you an application of our library that we've used to develop potent and functional antibodies against GLP-1R. In addition, we are actively using this platform to develop antibodies against a variety of GPCRs. If you would like to learn more about our library design, our discovery campaign, our discovery platform, or ways you can partner with us, please feel free to reach out to us or visit us at www.twistbiopharma.com. Thank you for watching this video. Have a good day.